In recent years, cancer therapy has made massive leaps thanks to new frontiers in immunotherapy. In 2011, a young girl by the name of Emily Whitehead woke up cancer-free as a result of experimental T-cell therapy she received at CHOP in a study led by Dr. Saven. After battling acute lymphoblastic leukemia for several years, Emily made a full recovery and has been cancer-free ever since. So what exactly does cancer immunotherapy involve? An understanding of this mechanism requires a comprehension of how our immune system works. Our bodies are equipped with a natural defense system against potential threats. Lymphocytes, a subtype of white blood cells, are an important aspect of this system. These cells come in many different types, including natural killer cells, B cells, and T cells. There are two major types of T cells, called CD4 and CD8. CD4 T cells are helper cells and send signals to other immune cells. CD8 T cells are cytotoxic cells, which secrete cytokines. Cytokines are small proteins which have anti-tumor and antiviral microbial effects. To prevent these T cells from becoming overactive and killing healthy cells, various inhibitory pathways help to limit the autoimmune response. T cells express several different inhibitory receptors on their surface, which are activated in response to cytokine release. Our focus will be programmed cell death 1, or PD-1, and how our understanding of it has allowed us to develop immunotherapeutic drugs for cancer patients. PD-1 functions as an effective brake on the T cell attack of a cell, whether it be healthy or cancerous. PD-L1 is a ligand expressed on the target cell that binds to PD-1, thereby inhibiting the T cell immune response. PD-L1 is overexpressed in cancer cells, effectively concealing them from T cells and promoting their proliferation. As seen in this figure, expression of PD-1 is positively correlated with progression of the tumor stage. This overexpression is regulated by a signaling pathway within the cancer cell, which is activated by interferon gamma released by the T cell. How did scientists explore the intricacies of T cells and their biochemical interactions? Flow cytometry is an experimental method used in the lab to characterize these cells during such studies. Physical properties such as size and surface proteins can resolve certain cell populations. This approach involves tagging PD-1 receptors with a mouse anti-human fluorescently conjugated antibody. The antibody, tagged by fluorescent dyes, presents the antigen for the receptor of interest. If the receptor is expressed on the cell, fluorescence intensity will be detected by the flow cytometer machine's laser at a particular wavelength. Studies of PD-1 expressing T cells have illuminated the nuances of this receptor-ligand pathway. PD-1 and PD-L1 are membrane proteins which fall into the class of immunoglobulin molecules. These proteins have specific V-set domains which resemble the antibody variable domain, and these are the means by which they interact. Basically, PD-1 and PD-L1 have beta sheets in their respective IgV domains, whose front faces undergo a hydrophobic interaction. This causes PD-1 to undergo significant rearrangements, as the residues of its CC loop change from open to closed conformation. These rearrangements are important for creating contacts that stabilize the interaction between the binding partners and allow for an induced fit. When engaged by the PD-L1 ligand, PD-1 inhibits its kinases involved in T cell activation through the dephosphorylation action of phosphatase SHP2. This effectively suppresses the activity of the T cell. Numerous therapies and drugs have been developed for the purpose of manipulating the PD-1-PD-L1 pathway. While the conventional approach to disrupting the PD-1 pathway would be to devise molecules that directly affect its active site, others have been made to act on separate regions on the molecule. One such drug is Opdivo, which makes use of the molecule nivolumab. As mentioned, PD-1 and PD-L1 binding involves the interaction of front beta sheet faces of their IgV domains. However, the binding of PD-1 to nivolumab is dominated by the end loop. The end loop of PD-1 contributes to 10 out of the 16 hydrogen bonds. Nivolumab contributes to the competitive inhibition of the PD-1 and PD-L1 complex, as the molecule sterically hinders PD-L1 binding. From the success of drugs such as Opdivo, we can see a horizon of possibilities for drug therapies that may target the PD-1-PD-L1 pathway, and ultimately other immune checkpoints. The potential applications of the PD-1-PD-L1 pathway are exciting, but still meet several challenges. Checkpoint blockade cancer immunotherapies have proven to be successful in treating blood cancers, as seen in Emily Whitehead's case. However, a minority of patients achieve long-term benefits from checkpoint blockade in solid tumor cancers. The tumor microenvironment is harsh, and functional significance in different cancers is still poorly understood. Furthermore, patient response to PD-1 blockade in particular varies, though studies have shown that a particularly dysfunctional T-cell subset can be identified by PD-1 expression and co-expression of multiple inhibitory receptors. This may assist in identifying patients likely to respond to inhibitory receptor-specific antibodies. Current research efforts are striving for a better understanding of these biochemical pathways, which may provide a framework for future personalized T-cell-based therapies. Through this biochemical concept, the future of cancer research has promising new frontiers, and stories like Emily Whitehead's will no longer be out of the ordinary.